Welcome to L121 Bio. We are about to begin what it means for an analysis of cell reproduction. And, we're going to go through Biology 2nd edition, OpenStax Volume 1, and we're going to look at how it is Mother Nature has figured out a way to take one cell, have it grow for a period of time, so that you ultimately end up making two smaller cells. Okay? And most of you remember that from earlier biology class, this will be a series of metabolic pathways that are going to occur inside this entire process. In Chapter 9, we spoke about what it means for signaling to happen maybe between cells or something like that. But one thing that Chapter 10 actually reinforces, though not directly, is what are the mechanisms that are required for intracellular communication, such that a cell, one, knows its size, and then two, what's actually happening inside the cytosol of a cell. I mean, you don't think about what's happening inside of you when you're digesting food. You don't think about what's happening inside of you when you're actually respiring gases in and out of your lungs through external respiration. Likewise, Mother Nature needed to figure out a series of steps with detectors that could determine the state of where a cell is at any given point in time. So that's actually what we're hoping to accomplish today. So how do we actually do it? Cell division has two functions. For multicellular organisms, you use cell division for growth, maintenance, and repair of cells. Okay? But for single-celled organisms, they use it primarily for reproduction. Now, reproduction we tend to cover inside Chapter 11, and I will bring pieces of meiosis into Chapter 10 so you can see the distinction at the end. But specifically speaking, what we're looking to do here is take some form of a cell, whatever the cell is, and we'll go through a series of either cell division, growth processes, the actual maintenance of what goes on from moment to moment, and then how do cells actually repair? Because this, when you think about it, is one of the quintessential questions. If you think about what happens to your skin when you actually cut it, what's going on? Most people will tell you they bleed. Some people will tell you there'll be a scab eventually. But the idea, of course, is you actually are repairing cells inside of that space. And it requires cell division and growth, as well as the maintenance of the materials, mainly the environment, in which those cells are growing into. So, the cell's DNA makes up what we call the genome, and inside prokaryotes, the genome consists of one double-stranded circular DNA molecule made up of a sugar phosphate backbone, has A's, T's, G's, and C's, but the idea is this. If I drew it for you, you're going to see inside the cartoon, here they have this sort of like squiggly, where the line represents a double-stranded piece of information, and that means on it, and within it, whoops, I didn't mean to undo that. I meant to change the color. So here's that double-stranded squiggly thing. What it means, of course, is that within it, let's say that there's a segment here that actually encodes for specific proteins, and maybe there's another one down here. But the idea, of course, is then the regulation and or something simple, like duplicating it, is quite elegant. I mean, in the sense that it's a pretty straightforward system where you end up then just making a copy of this. They reside in what's called the nucleoid structure. We don't tend to think too much about them, even though I've had you hopefully you went and actually looked up that quora sensing, so you can see how quora sensing regulates transcription and translation. Okay? It's a really, when you think about how we move from a single-celled organism to multicellular organisms, what that means is there's some adaptation where you're signaling between cells in such a manner that it's coordinated. Now you can also put other pieces of genetic information inside of here, and they are what we call plasmids. And they are not necessarily for normal growth, but they also have important information. And exchange of information between plasmids is actually how you get gene transfers. Let's put a bacteria over here. Okay, we'll put some pili on the outside of it. Okay. And what will happen is you literally send plasmids between pili so that you actually mix up the genome between these organisms. And that allows for a heterogeneity of gene expression and mutations found inside those sequences. That's one of the mechanisms that allows organisms such as this to survive in multiple different environments for their period of time they're going to survive. So we'll get rid of that. The eukaryotic genome, here represented, consists of, so this is actually a plant cell. These are a series of plant cells undergoing different stages of growth. Consists of a double-stranded DNA molecule, just like we saw back in, I want to say chapter 4. It might have been chapter 3. But the idea, of course, is depending upon where we are in a cell's life cycle. Okay, where we are inside a cell's life cycle, you'll see I draw it a specific way. Your textbook's going to draw it in a different way, but whatever, they're all telling the same thing. Where you're going to break apart what's going on inside of a cell, doing the normal job that it's actually doing, primarily is here inside a state we're going to call interphase, and then we have this thing that's mitosis down here. You all remember mitosis. In fact, it's going to be part of your lab. Mitosis, right? Fantastic. But what I want to make sure people understand here is chromosomes. Okay? That's the, let's go A, T, T, A. Or the blue, you've got the sugar phosphate backbones. Here you have the nucleotides. Okay? And remember, they're encoding specific pieces of information through the complementary base pairing that's held together through hydrogen bonding. And what's going on here is when Mother Nature needs to package double-stranded structures, what she ends up doing is she makes these things that are called chromosomes. And what's really important to understand is that in chromosomes, when you actually see the condensed material like you do here, whatever information is on those structures, it's not accessible for utility. So that means the only time a cell is actually undergoing maintenance and or growth and or replication processes is when it is uncondensed and not in the chromosomal shape. So here are 23 pair of chromosomes, and this is what literally when, how would I say this? So histopathologists will stain the cells of whatever, namely, let's pull a cell out of your body someplace, epithelial cell, right? And they go and they do a chromosomal smear, and what they do is they look for how you separate these molecules by size, and what you end up with, of course, are chromosomes inside the cell nuclei are going to vary. The actual number varies between each organism. So humans have 23 pair. It's 22 somatic and 1 pair of sex chromosomes. Now, what this means, so what you actually say X and Y here, so they separated 1 through 7, 8 through 15, 16 through 22, and here is the X and the Y chromosome here. And what happens, of course, is the chromosome number is consistent, but they may vary depending upon the developmental stage or a specific cell type. You're going to see we will not discuss that much inside of this class. But what I want you to see here is each of these banding profiles represents locations of genes that are expressed from either one chromosomal arm or the other chromosomal arm. And you'll see how that works a little bit later on. Remember I said those somatic cells are diploid structures, and what we do inside chapter 11 is you think about, even though we don't cover it inside this class, you think about how it is you actually make haploid cells. Because unfortunately, most of the cells inside of your body are diploid, which means there are those two copies we saw in the previous here, one mom, one dad. And what happens, of course, each one of these, one gene on one allele will be expressed, the other will be silenced. But now, what Mother Nature needs to do to increase genetic diversity is she'll make a series of structures that we call gametes. And those gametes are haploid cells. They have one half the chromosomes that you would normally find inside of a diploid cell structure. And that's when we will end up talking about what it means for sexual reproduction, right? When you actually have the meeting of, say, a spermatocyte and a secondary oocyte, right? Where you actually have the fusion of the pronuclei that has the 23 chromosomes over here, and over here you'll have 23 chromosomes there, and those 23 chromosomes will meet at fertilization, and then you end up with where you started off as a living human being. A one-celled structure that is now diploid. But it's important to understand that the egg here and the sperm there were once haploid cells. And that meant that they had 23 chromosomes. So now, why do we actually care about this? Well, we care about this because we arrange the chromosomes by size, and this is the karyotyping I was telling you about, and they're arranged as heterologous pairs. But we organize it in terms of, if this is the DNA double helix here, and you can see the sugar phosphate backbones, and here's their representation of A's, T's, G's, and C's. When we need to get it to all fit inside of a nucleus, what we do is we wrap it around these structures that are going to be called histones. Okay? And what's going to happen is once you've done that, those histones will then coil around themselves so that you actually make up what's called a nucleosome. So that's the coiled structure here. And then you finally condense this, okay, such that you end up with what we would call condensed chromatin matter. And that's how you actually end up making a chromosome. But it's interesting. What I want to make sure you understand is it's the uncondensed form of DNA that is actually the active form of DNA. The only time we actually see this is when we are going to move it. Okay? It's the only time you actually see condensed nuclear material. So we have to package it. And I just told you essentially how we do this. And we create these things that are called linker DNA structures. And this allows for efficient packaging. And there's a little movie you guys can go watch here when you get the free time to do what you want to do. But the idea, of course, is this is a representation of what we think actually happens. All of these proteins have been separated out by a series of extraction steps. And the important thing is this, at least way back when this was first discovered, we used to have a journal club about it. And what we would talk about is what Mother Nature could be designing in doing this. And most definitely, her goal here is to protect the integrity of that DNA. And if you figure out wrapping around a alpha helical structure around itself, what it does is it ensures protection from different forces. Now, why we care about this, of course, is something completely different. All of the information on those individual pairs of chromosomes that, when unfurled, are biologically active, is accessible here. Let's change colors for you. Primarily during this interphase piece, which is why Mother Nature does this. And what she does is she creates these series of mechanisms that allow for things to occur. And that is, growth will occur. Should we need to replicate the genome, that occurs. And then we check to make sure that everything was done properly. So the pattern is cell growth, replication of genome, and then continuation of growth. And then, because this has all happened, what you do is you then will separate the replicated materials and then create those two new formed cells. Notice textbooks call them daughter cells. Okay? And this means that they are distinct from one another. They may have arisen from the same one cell, but by the time they are finished, they are unique from one another. Okay? So I want to cover a few more here before we finish, a few more slides to make sure you get the information properly. And what's going to happen, of course, is here, the cell cycle has two major phases. And in blue here, we're going to call this interphase. Okay, and that's preparation for cell division. And then the mitotic phase. And this is the focus of your lab I mentioned a moment ago, where you're going to literally go find those individual structures on your own. What I want to make sure you understand is we can break this into these parts, and here's what you know is actually happening. Gap 1 is when the cell is biochemically active. It's doing all of its responsibilities. In S phase, we're going to literally use a molecule called DNA polymerase and a complex of other molecules, and what happens is it replicates an identical copy of DNA molecules, such that you end up with sister chromatid structures that are joined at the centromere. And the purpose of this, of course, is they're going to use, inside animal cells at least, those centrosomes that we've just engineered into this to actually help associate the movement, okay? To make sure that we separate the chromosome numbers properly. And in G2, this last piece here, okay, the second gap is energy is replenished, organelles reproduce, meaning we will duplicate everything that we need, and the cytoskeleton literally begins to break down. And it's being redistributed between the two poles of this one cell. So that when we actually finish mitosis, which you see up here, okay, you will now be able to have two functioning cells. Now, karyokinesis is also called mitosis, or the first step. And what's going to happen, of course, is these are the individual stages where we're going to pull the genetic material apart. You're going to see how that happens in a minute. And then in the end, the cytokinesis piece is when you actually split that one cell into two completely distinct cells here. Okay. Now, inside every single textbook, you're going to find something that looks sort of like this, or mitosis or karyokinesis is the separation where you have the nuclear envelope breakdown and the condensation of the nuclear materials, meaning completed condensation of the replicated genome. And then in pro-metaphase, we're going to start aligning the cytoskeletal structures to head towards the midpoint of the cell and align now all of, in metaphase, the replicated chromosomal structures along what we'll end up calling the metaphase plate. During anaphase, what happens is literally you see all these microtubule proteins. They've gone fishing and they've grabbed onto one half of that chromosome. And they begin to pull back towards the poles of these cells, going towards the centrosomes. Now, as we move from anaphase into telophase, you're going to see that, look, we have an equal distribution of mitochondria, Golgi apparatus, all that sort of stuff. So the nuclear envelope begins to reform, and we begin to put an actin cinching plate through here, so that during cytokinesis, a cleavage furrow is going to form, and that cleavage furrow is going to allow for the complete separation now of two functional cells. Now, I'm not going to read through all these different pieces here, but now, you know what? I really should. I've got a minute or so here. Mitosis begins with prophase, and you can see what it's doing as the nucleolus disappears, centrosomes begin to migrate. All of these things have to happen in a stepwise manner. And in this thing that we call the kinetochore here, notice what's actually happening. These are the centromeres. We're attaching mitotic spindle microtubules here. And this is actually how we anchor and direct where the sister chromatids are going to actually end up traveling to. During metaphase, we line them up, and the sister chromatids remain attached by a specific protein, and that protein is named cohesin or cohesion. Okay? We're going to break apart that, degenerating the actual chromosomes now we're going to pull towards opposite poles. Ultimately, we get to telophase, we reform the nuclear envelope, they decondense, and now the cell can begin to actually undergo cytokinesis, where you put in an actin-based contractile ring, okay, versus a plant cell, which will use Golgi vesicles to separate the cell into two separate cells here. So how does this all happen? Well, that's going to be the second piece, but I want to give you a sort of what Mother Nature is doing in the next set of slides. So I'm going to pause here and just say that Mother Nature has a series of regulation steps, and we call these checkpoints.